Welcome everyone to today's webinar. Our focus will specifically be on Naflsim use cases within drug development, and several new case studies will be shared with our expert panelists. Joining me is Chief Scientist Scott Q. Seiler and Scientist Christina Batista and Zach Kenz. Thank you all for being here. Quick housekeeping notes. We take your privacy rights very seriously, and by attending our event today or participating in the Q&A session, you're allowing us to contact you for follow-up. Your participation is highly encouraged. Remember, innovation begins with questions, so don't be shy. Please use the questions panel on your dashboard to submit your inquiries at any time during the presentation. And if you need assistance during the webinar, use the hand raise icon and our staff will assist you. Without further delay, I turn things over to Scott. Scott, please take it away.
Thank you, Arlene. So welcome everybody, the NAFLDsym application showcase. Christina, Zach, and I will walk you through a series of applications, as well as give the opportunity to, for those who don't know much about it, a brief introduction to NAFLDsym in addition to the actual applications. Our legal advice is always omnipresent, and I allow you, here I ramble on while you get the opportunity to read the disclaimer. Now we can proceed with kind of the 40,000-foot view of NAFLD, NASH, and where NAFLDsym fits. So for those of you who are familiar with the NASH space, this will probably be a little bit repetitive. For those of you who aren't, it's an interesting introduction. NASH is a progressive disease with mortality rates being linked to the severity of the disease. And equally as kind of concerning is the fact that the incidence worldwide of NAFLD and NASH is growing tremendously. Let me just take a step back. Non-alcoholic fatty liver disease and non-alcoholic steatohepatitis are the acronyms NAFLD and NASH. NASH tends to describe kind of the more severe portion of the disease, and NAFLD tends to describe the less severe. Nonetheless, all these patients with both NAFLD and NASH, there are tons of them, unfortunately. Also, it's worth pointing out there are currently no pharmacologic options for treating these patients. So what we have is just a substantial opportunity to improve the health of a vast number of folks across the world. To that end, there are numerous compounds in development across industry. And with fits and starts, 2 steps forward, 1 step back, and not unlike drug development in general. But in particular, within the last year or so, we've had some, within the NASH drug development area, some setbacks and terminations of programs that really have kind of, I would say, shaken up the landscape a bit. And not only has it shaken up the landscape, it really then points towards, I think, a justification for us and our motivation in developing NAFLDsym in the first place. So it's a quantitative systems pharmacology, mechanistic mathematical model of both NAFLD and NASH that is designed to support drug development. As was the case with most QSP models, it can be applied in numerous ways. Of course, you'll be hearing some very specific examples of that here today. And we have been using it quite a bit. We've got a fair amount of flight time with it. I think we've now evaluated almost 25 different targets and/or compounds with NAFLD and NASH, working with a variety of large pharma companies, those listed here on the slide, as well as others who are still remaining behind the proprietary wall. We have our own custom software that's available for licensing, as well as services where you can let the expert modeler, so to speak, run the simulations and do the analyses on your behalf. Before I go any further, I believe there's a poll question, yes?
Yes, there is. Are you ready to learn about Naflsim? We'll give you just a few seconds here. All right, an overwhelming yes. Scott, please continue.
Great. Okay, so then the agenda is, again, a brief introduction into NAFLDsym, but really the meat and potatoes, if you will, of the presentation today is focusing on specific examples The first is focus on the simulations of Senec-Revirox lack of efficacy and the ability of Naflsim to predict this. Simulations of BFKB8488A, providing a mechanism of understanding, mechanism of action understanding, as well as supporting dosing paradigm selection. And then finally, we'll conclude with an impact of lifestyle and the impact of weight loss and weight gain potentially on predicted efficacy. Of course, we'll conclude with questions, and hopefully you'll get an opportunity to ask those at any point during the session, but we'll answer them at the end. So at Simulations Plus, fantastic sciences all across the organization. One thing that I think is really important to point out is that we really do have expertise in a wide array of different modeling approaches. Of course, cheminformatics, PBPK modeling, pharmacometrics, and within our division, the DILIsym Services division, the QSP and QST modeling. Pulling together all this wide variety of expertise, we have a variety of software offerings and service offerings that are applicable all throughout the pipeline, from the earliest discovery to FIH to phase 3 to even post-approval. Our modeling approaches are pulled in, again, anywhere throughout the pipeline. If we zoom in on the QSP QST section, this is perhaps the youngest sibling, if you will, of the modeling family within Simulations Plus. QSP modeling, I'd say, really got its feet under it kind of at the beginning of the 2000s, almost exactly year 2000. And it was during that period of time that really the methodology in kind of that early to mid 2000s, the methodology for QSP and QST modeling was really being worked out, honed, and refined. In fact, it's worth noting that several members of our DILIsym Services staff were actually involved in refining and establishing those methodologies. At this point, it's now spinning forward some 20 some odd years, we're now to the point where the methodologies are really well worked out, and we understand that you really need to understand the literature, understand the scope of the disease, hence the scope of the model, represent those key aspects, captures with your simulations the behaviors, build confidence within not only the modelers but the larger project team, explore knowledge gaps, and then the interaction with the experiments to support study design, gather data, and then kind of keep iterating, improving both the experimental paradigms, the data collection efforts, as well as the modeling efforts. In fact, as you might have guessed, DILIsym Services does in fact employ these very methodologies. Again, we look into the literature, into the science, into the various types of data to make sure that we understand how the connections are formed within the disease state, within the relevant pathophysiology or biochemistry. We then, of course, apply our mathematical equations to those and then run the simulations. And it's also important to note, very important, that it's not just about the disease, it's about understanding the treatment and the response to treatment for those simulated patients with disease. Again, the focus is all come together, enabling us to then develop models of quantitative systems toxicology or pharmacology. The difference being, of course, with toxicology, we're predicting safety, whereas with quantitative systems pharmacology, the focus is more so on efficacy, perhaps. Some of our offerings are listed here: RENAsym, DILIsym in the QST space, with RADAsym, IPFsym, and NAFLDsym in the QSP space. In addition to these software offerings, one other offering, I guess you would say, that is occurring with greater frequency is us developing, DILIsym Services developing custom QSP, QST models for industry, for various partners within industry. In these cases, a model doesn't exist for a specific disease area in order to answer a specific question. And we certainly have a fantastic group of practitioners of the art, so to speak, of QSP and QST modelers. And so we're being engaged more with ever increasing frequency to develop these custom models. I point this out largely because within the modeling community, sometimes we feel like we don't have quite enough tools. And I'm now offering our services to help build some of those tools to support the modeling community. Again, so now if we home back in on the DILIsym services division, as you might imagine, developing all these different disease models in these different areas has allowed us to establish deep knowledge in multiple disease areas, as well as the toxicology realm. And all of our models are available for licensing and or consulting projects, in addition to that custom QSP model development to which I just referred. We'll focus today, of course, on NAFLDsym. And before we dive into that a little more deeply, let me just as well point out that, of course, the DILIsym Services Division, all of Simulations Plus, really follows the innovation, the respect, the integrity, the commitment. The same values across the organization are applied to ensure the standard of the work and the standard of the relationships. Okay, so a brief overview of NAFLDsym V2A. Basically, we have found that NAFLD and NASH are really broken up into kind of 3 or 4 distinct areas, and we've kind of colored and coded it for your convenience in this diagram. This diagram describes some of the major players. It is not all encompassing of everything that's in the model, but in white, we have the major players, if you will, again, of the steatosis, or the lipid dysregulation within the liver. In red, it is the lipotoxicity, how the lipids then can affect the health and steer the hepatocytes towards death. In blue is the inflammation and how those dying hepatocytes can activate inflammatory pathways within the liver as well. And then finally, how those pro-inflammatory pathways then have the ability to interact with the pro-fibrotic pathways in green, activating the stellate cells and the propensity for the synthesis of collagen and extracellular matrix proteins, so that really we have kind of each of those 4 sections very well represented, not just diagrammatically, but mathematically, and as you'll see as we go forward, the ability to actually accurately predict and help increase the understanding of each of these specific areas. Each of these submodels, in addition, there's one other, which is weight gain and weight loss. This is important as one of the examples we'll show later, but in particular, weight gain and weight loss and the mechanistic influence that it applies on these pathways is captured within NAFLDsym. All of this is representing human clinical patients, and it's been based almost entirely on clinical literature. We do have numerous simulated patients to capture the heterogeneity that is certainly in effect in this patient population. And of course, it provides the ability to predict the responses in those sort of clinical environments. As far as the outputs go, this is, generally speaking, the primary outputs that regulators really pay attention to are all as well outputs of NAFLDsym. If we focus initially on the histology measurements, so there's the NAFLD activity score and its components. Those are all captured in NAFLDsym, as well as the fibrosis stage. Those are probably the most heavily emphasized by regulators, although certainly for later stage compounds, that's the case. As we move over into kind of earlier in the pipeline, imaging modalities like the MRI, the ability to measure liver fat percentage, is more heavily emphasized. Again, NAFLDsym has the ability to predict changes or predict liver fat percentages, in addition to some circulating biomarkers like ALT as well, for instance, and plasma ProC3. All of these listed here, in addition to pretty much every equation, every state variable within NAFLDsym is as well viewable. So you can gain a much broader understanding, but these are those that are perhaps most clinically relevant. With respect to our simulated populations, or what we would call SimPops, we're applying the methodologies that were developed several years back, wherein we are looking at each of those specific areas of the pathophysiology, and then kind of triangulating between the model and the relevant literature to establish the range of responses, the range of the kind of the variability within each of these categories, and then doing the combinatorics by assigning parameters within each of those ranges to generate a potential simulated patient. Interestingly, that of course establishes a pathophysiologic variability by definition. It also, interestingly, kind of filters into some very distinct buckets of clinical variability as well. We take these potential simulated patients, and then we compare them with a multitude of clinical data across numerous axes all at the same time to establish a validation of these simulated patients. And that, of course, catches them in the disease state in order to get a sense of their response to treatment. The standard approach is to apply standards of care to the simulated patients, the SimPops. In this case, there is, as I mentioned before, no pharmacologic option available. So we're applying the current standard of care, which is weight loss. And so all the simulated patients are subjected to weight loss, and we ensure that the response is, in fact, appropriate. So that kind of concludes the brief introduction of NAFLD and NASH and our division, DILIsym Services. At this point, we have one more, or well, another, I should say, poll question. So Arlene?
Yes, and this one is a fun one. So what major pathophysiologic aspects of NASH are represented in NAFLDsym? Please select one, and this is, we promise not to share these answers, so feel that you can answer them away. No one will be called on except maybe Scott, and we'll give you just a couple of seconds here. All right. Scott, back to you.
Okay, thank you, Arlene. So with that, actually, I get to hand the baton over to my colleague, Christina Batista, to walk us through the next section. Go ahead, Christina.
Great, thanks, Scott. So in this section, we're going to look at our simulations that predicted really lack of efficacy for Senecor within NAFLDsym. Next. So hopefully within this section, I will convince you that if Senecor had been investigated within NAFLDsym, we could have looked into the clinical potential prior to Senecor being put into clinical trials. So with that, we'll just jump right in. So as a brief introduction for Senecor, back one slide, please. As a brief introduction for Senecor or CBC, as you'll see throughout the slides, it's a dual CCR2, CCR5 antagonist, and it was being used to treat adults with NAFLD-NASH. It's thought to act by inhibiting the recruitment of macrophages, as well as inhibiting activation of hepatic stellate cells. So by inhibiting recruitment of macrophages, you're inhibiting cells, immune cells flowing into the liver, also inhibiting the mediators that are being produced by some of those immune cells. By inhibiting the activation of hepatic stellate cells, you're inhibiting the cells that are synthesizing collagen and depositing collagen, so thereby thought to inhibit fibrotic worsening. So really, Senecor was thought to have potent anti-inflammatory and anti-fibrotic properties. With that being said, Senecor had been progressing through clinical trials, so we sought to represent it within NAFLDsym, and our simulations completed in late 2020. As the title of this section indicated, NAFLDsym predicted really minimal efficacy in our simulations, and we had designed a NASH cohort specifically for this to be consistent with the characteristics of the clinical patients in the Senecor trials that were ongoing. Fast forward a few months into January of 2021, this year, and actually the phase 3 clinical trials, the Aurora study for Senecor was terminated, and you can see the screenshot on the right, indicating that it was terminated and it was cited as due to lack of efficacy. So this was really nice validation for the results that we had just produced a few months prior. Not as exciting for the NAFLD-NASH treatment community, but really nice to align with the results that we had made prior to this. Next. So when we started incorporating Senecor within NAFLDsym, we referenced a lot of the data that had already been published from earlier clinical trials. So we focused a lot on the CEMTAR data that had been published. The year 1 study had been published by Friedman and others in 2018. And although it indicated that Senecor didn't meet the primary outcome, which was at least a 2-point improvement in that NAS, the NAFLD activity score, with no fibrotic worsening, they did say that they achieved significance with regard to the secondary outcome. And so that's at least a 1-stage improvement in fibrosis score with no NASH worsening. They followed this study up by their final 2-year analysis published in the Rat Zoo paper in 2020, where unfortunately they concluded that Senecor didn't maintain significance compared to the placebo group. And so I'm showing actually on the right in the figure we're looking at that secondary outcome, an improvement in fibrosis by at least one stage with no worsening of NASH. And I'm indicating the placebo group with the open bars and the senochrivirac treated in the shaded bars. So you see there's really no distinguishing the senochrivirac from the placebo group. So they didn't maintain any significance from placebo, unfortunately, with the senochrivirac group. So this really gave us motivation to see if we could capture this within NAFLDsym itself. Next. As we discussed, CCR2, CCR5 antagonists are thought to inhibit recruitment of immune cells into the liver, as well as inhibit activation of some of those stellate cells. So we're putting in, those pathways exist within NAFLDsym, and we're putting in regulation on that pathway due to our CCR2/5 antagonist. Next. Looking to parameterize this effect of senochrivirac on these two pathways, we went into the literature. So from published literature, we're able to derive parameter values for the effect of senochrivirac on these different mechanisms of action. So on the left-hand side of this screen, we're looking at data from the Kruger 2018 paper, where they're investigating the effect of senochrivirac on activation of hepatic stellate cells. So they're taking human hepatic stellate cells, activating them with TGF-beta, and then adding various amounts of concentrations for senochrivirac as well, and measuring the change in activation for these stellate cells. The data do indicate a maximum inhibition of about 80%, and we see that the inhibitory effect of senochrivirac becomes engaged at higher concentrations. We're neglecting those potential stimulatory responses for senochrivirac at the low concentrations. These low concentration results are not significantly different from the TGF-beta alone treated results, so we are seeing a maximum inhibition of 80%. So we can use this data to parameterize the values for the effect of senochrivirac on activation of hepatic stellate cells. On the right-hand side of the screen, we're looking at data from the D'Antonio 2018 paper, where they're investigating not only senochrivirac, but some other CCR2 and a CCR5 antagonist on monocyte migratory factor. So you're seeing a 40% reduction in the amount of cells being recruited, and we're indicating this not quite as fleshed out. You don't have quite as many concentrations to compare here, but we're assuming this as a maximum inhibition of 40%, and this inhibition occurred at a concentration of 1 micromolar or 0.696 microgram per mil. So these data will provide an idea for the mechanism of action for senochrivirac and the potency of it. Next. Not only do we need to take into account the potency, we also need to know exactly what exposure we're seeing within the liver and see if that's enough to drive the effects. So in order to answer that question, we derived a PBPK model for senochrivirac. We developed this model and optimized it based on data from healthy volunteers in clinics. And we focused on the 150 mg QD dose. That's the dose that was being used in the Centaur trial. You can see in the figure on the top, the red line shows our simulated plasma profile predictions from our PBPK model. And it's being compared to just one of the data sets that we had used in optimizing this model, the Lefebvre 2013 data set. Again, these are healthy individuals, and we've optimized our model to this plasma profile from a source of data sets. In constructing the model, we did use parameter values from the literature wherever possible. Of course, not all of those parameters were available within the literature. So for some parameters, we estimated them using ADMET predictor, which estimates the values based on the chemical structure of senochrivirac. And for other parameters where we weren't able to use ADMET predictor and there weren't literature values, we did optimize them to allow us to match the plasma profiles following senochrivirac administration. This PBK model was designed really in order to give us an accurate prediction or an estimation for the liver sinusoidal blood concentration, and that's the figure shown in the bottom. We're showing it for the 3 different zones within the liver, the central lobular, midlobular, and periportal. And these concentrations are then exported and used within NAFLDsym to drive the effects of senochrivirac on stellate cell activation and monocyte recruitment. It's thought that, we think the liver sinusoidal compartment is where senochrivirac has the opportunity to interact with these different cell types. And so that's why we chose this concentration to drive our effects within NAFLDsym. Next. The last piece that we had to start developing in order to run our simulations is we wanted to look into our cohorts. So Scott had mentioned, we have 1,700 individuals to choose from. That's a lot of individuals for us to simulate. We pulled out a select few here and we've classified them based on their fibrosis score. So we have an F1, F2, and F3 SIM cohort within NAFLDsym, and we're showing their baseline characteristics here. You see there's lots of variability to capture what you would see in a clinical trial. And on the far right, we're actually comparing them to the Centaur data that was published in the Freeman 2018 paper. And you can see, well, there's only a few outputs that we compare directly with NAFLDsym with our SIM cohorts. We are doing a nice job of capturing the variability that was seen in that Centaur trial. So this puts us on an even playing field, knowing that the individuals we're simulating are representative of that Centaur clinical trial and patient cohort as well. Next. So now that we have all the pieces together, we are well suited to simulate Senecrevirac within NAFLDsym and kind of under the exact same conditions as they saw with the Centaur study. So we're going to administer Senecrevirac daily, 150 milligrams per day for 2 years, the same length as the Centaur study. We're using those Senecrevirac informed parameter values for the effects on monocyte macrophage recruitment and hepatic stellate cell activation. And then those effects are going to be further driven by the exposure that you see in the liver sinusoidal blood compartments. And again, we're simulating these in the F1, F2, F3 SIM cohorts, which aligns with the cohorts that were used in the Centaur clinical trials. So with all the setup being put in place, we can jump to the next slide and look at some of our results coming off of NAFLDsym. So as Scott mentioned, some of these histological outputs are the most relevant for these NAFLD-NASH studies. And so we're looking at 2 of them here, just to orient you to these slides. The top and bottom row are different outcomes. So the top row here is the NAFLD activity score. The bottom row is the fibrosis stage. And then from left to right, we have our different cohorts within NAFLDsym, F1, F2, F3, from left to right. So again, focusing on that top row for our NAS over the 104-week simulation, we're not seeing any changes, right? Our individuals are it looks basically flatlined across our simulation results. We're not getting any improvement in NAS with Senecrevirac. Shifting to the bottom where we're looking at fibrosis stage, if you look on the far right, we are seeing some improvement in F3. We also see at least 1 individual worsening. But so Senecrevirac does seem to have some impact, at least on fibrosis stage, particularly in the F3 cohort here. We can move to the next slide and look at some collagen outcomes in the same cohorts, same simulation results. In the top, we're looking at collagen percent area, which is really indicating the total amount of collagen in the liver. And it's a similar story to what we saw on the previous slide. You're seeing minimal to no improvement with Senecrevirac in any of our cohorts here. Shifting focus to the bottom where we're looking at plasma Pro-C3, it's a biomarker for the synthesis of type 3 collagen within the liver. And again, we're seeing maybe minor improvements in the F3 cohort if you really squint, but overall, no large improvements or significant improvements with Senecrevirac treatment. The last results I wanted to share in this format are looking at the potency of Senecrevirac. So we talked about Senecrevirac inhibiting recruitment of macrophages and inhibiting activation of stellate cells. So on the top, we're looking at the effect of Senecrevirac on macrophages. Here we're looking at total portal macrophages, and we can actually see a reduction in portal macrophages throughout the course of the simulation, indicating that Senecrevirac is having an effect on macrophages as intended. However, the same can't be said for activated hepatic stellate cells. If you shift your focus to the bottom 3 figures, you can kind of see a slight decline, especially if you look at the F3 cohort, but it's very minimal, especially compared to the effect that Senecrevirac has on portal macrophages. Putting all this together in kind of a quantitative manner on this slide, we can compare those primary and secondary outcomes. So here in the far right column, I'm looking at the primary outcome. From our SIM cohorts, we didn't have any improvement in NAS in any of our F1, F2, or F3 SIM cohorts. And while this wasn't exactly reported in RATZU 2020, they did state that they didn't meet the primary outcomes significantly. So our simulation results align quite well with their data. Shifting to the column 1 to the left, we're looking at that secondary outcome, that at least 1 stage fibrosis improvement with no worsening of NASH. We do have some individuals meeting that criteria within our F3 cohort, but it's pretty minimal compared to the size of our entire simulated population. Comparing that to the data from the Centaur study, well, they did see improvement. They saw it in both the Senecrevirac treated and the placebo treated groups, and they weren't distinguishable. So our results really align with the idea that Senecrevirac had a minimal if any, improvement on fibrotic outcomes within the population. Next. So just to wrap things up, I hope I made it clear our 2-year simulations for Senecrevirac really predicted no or minimal improvement with regards to NAS and fibrotic outcomes. And this was entirely consistent with the 2-year Centaur results that had been published and with the early termination for phase 3 Aurora study that was cited due to lack of efficacy. We had used different pieces of available literature in designing our model, as well as in constructing the PBPK model, and we had designed sim cohorts that specifically represented the individual variability that they saw in the clinical trials for Senecrevirac. So had Senecrevirac been investigated in NAFLDsym prior to the clinical trial, the clinical potential could have been explored, thus saving time, money, resources, and maybe shifting drug development focuses elsewhere. So I'll conclude with this section here, and I think it's time for us to have another poll.
Thank you, Christina. Here we go. Can NAFLDsym predict changes to NAS and fibrosis score? Go ahead and select your options, and on deck will be Zach. Okay, Zach, you're up to bat.
All right, thank you. So the next study, we can go to the following slide, is looking at using NAFLDsym to examine the mechanism of action for a particular treatment, and then using that to help inform the selection of dosing paradigms. And so for this study or case study that we're looking at, BFKB8488A, which is an agonist anti-FGFR1 beta-klotho bispecific antibody, is under development still to treat patients with NASH. And so what we were trying to do here was not only look at modeling within NAFLDsym, looking at efficacy, but also help better understand the particular mechanism of action, which is related to an increase in serum adiponectin associated with the treatment. And so if we can better understand what will happen with serum adiponectin increases and the subsequent impacts on the liver, as well as some other mechanisms of action that I'll discuss, we can inform what's happening due to treatment, and then also the particular efficacy under some different dosing paradigms that Genentech was looking at. And so when we represented this compound in NAFLDsym, we got a few different results that we'll go through. So this poster was represented at the liver meeting in 2020, and we'll call out a few particular things, particular key aspects of this poster that we'll walk through. So if we go to the next slide, we'll look at the particular mechanisms of action here. So starting in the lower middle figure here, it's a FGF21 sort of analog. It acts in a similar manner as FGF21 on the adipose tissue. So the receptors there, and the two main effects are, as noted here, increasing adiponectin secretion and also increasing uptake from the plasma of plasma triglycerides into the adipose. And so that increase in adiponectin mediates then through hepatic receptors enhancement of hepatocellular AMP kinase. And so if you follow those green arrows into the liver, that has downstream effects of decreasing de novo lipogenesis, which also can increase hepatic secretion of VLDL triglycerides, decreasing esterification of fatty acids, and then also increasing fatty acid oxidation. And all those effects together can ultimately decrease liver triglycerides. And so the goal is to see an efficacious result of decreasing liver fat. And you can ultimately see there's a lot of different effects, and by mechanistically modeling the effects of this particular compound, we can show where the different mass might go. We could look at accumulations of fatty acids at different places, what's happening in the adipose, and see what we think is actually going on with this compound, and match it with what we're seeing in the clinical cohort. So those are the key mechanisms of action, and again, adiponectin is one of the key aspects here that we want to look at in these different dosing paradigms. So the Genentech was looking at subcutaneous administration at 50, 75, and 100 milligrams every 2 weeks, and then also an every 4-week 250 milligram dosing paradigm. And we did simulations for 12 weeks here. And we ultimately split this phase 1b clinical data into 2 cases. So we looked at a low dose every 2 weeks and then that every 4-week data to do our initial calibration, and then validated on the 75 and 100 milligram every 2-week data. So we could actually see sort of based on that phase 1b data, we could do a little bit of validation of our modeling that was going on. And then also, since adiponectin was a key mediator of effects, we did simulate with and without that adiponectin increase to test that key mechanism of action hypothesis for BFKB 8488A. And then another key aspect, and just like what Christina was discussing, we did look at asym cohorts within NAFLDsym of a size 42, which was similar to the cohort that they were working with that had characteristics similar to the clinical cohort, which again is important when you're trying to talk about matching your mechanistic model with what's going on in the clinic. So this gets us from our mechanisms and gets us clinical cohort and clinical dosing paradigms that are matching between the model and what they're seeing in terms of actual patients. And so if we bring this forward, then we can get to looking at the actual results. And so we'll start from left to right. So here, if we want to, kind of dig into what's actually going on and what was very informative for Genentech and bridging from phase 1b into phase 2. The far left figure is looking at the adiponectin changes. And so this is changes from baseline in their patients over the 12-week period. So in all of these figures, the black circles are their clinical data. The red open circles are the simulation results parameterized with the adiponectin increase, so what we think the mechanism of action is. And then the pink filled in circles are parameterized without that adiponectin increase. And so in the far left figure, if you follow the red open circles, you can see we've parameterized a dose-dependent increase in adiponectin, and that generally corresponds with the clinical data. That third clinical data set, there were some potential issues there, and that's why the clinical data and our simulations aren't as in alignment as the other cases. And then in the middle figure, we inherit some of that nice pathophysiologic variability that Scott discussed early on from the SymPops. And so we leverage those adiponectin changes to mediate the liver fat effects. And those have been parameterized to attain the simulated results and simulated variability that if you compare the red symbols in each of these dosing cases that and those doses match up with the 4 columns in the adiponectin changes, those variability in the simulated patients match up reasonably well with the clinical data, so comparing red to black. And then the other key aspect here is if you look at the pink circles that are along the 0% liver fat change, if the adiponectin increase isn't present, then there's no change in liver fat because there's nothing to mediate those changes. So this is, a way to kind of confirm our hypothesis that there's no liver fat changes without that adiponectin mediation. But on the far right, if you look at simulated patients versus the plasma triglyceride changes, that's through a different well, it's through that direct effect in the adipose where there's plasma triglyceride uptake. And so there are still, even without the adiponectin increase, there are still plasma triglyceride changes in the simulated patients, the pink and the red. And there's still some variability there that's reasonably in line with the clinical data. So in patients, there's a wide range of variability in plasma triglycerides, but we do expect that as the dose increases and exposure increases, which is roughly what's happening from left to right, that we would see a stronger change in plasma triglycerides. So overall, this was a very good demonstration of the mechanistic representation in NAFLDsym and very informative in terms of dose response back to Genentech. So if we go to the next slide, we accurately predicted the liver fat reductions and simulated the mechanisms of action, and we're able to balance all these different effects. And then also highlight this key mechanistic role for adiponectin in BFKB 8488A in terms of that role in driving those liver fat effects. And then this ultimately informed the dosing paradigm selection, and there's an ongoing phase 2 trial that this helped inform some of that process. So yeah, and I think there is a poll question as well at the end of this section.
Yes, you're right. Does your organization have a compound in clinical studies where the primary endpoint is change to liver fat? We'll give you a few seconds here. And if you haven't already done so, feel free to pop your question into the questions panel. And in a few minutes, we'll be starting the Q&A. So I'll go ahead and close out the poll now. And back to the panel.
Okay, that's me. I'm up. I'm Scott again. So let me wrap it up by describing at least one aspect of lifestyle that can impact potentially the predictions of efficacy due to pharmacologic effects. So it's really quite interesting. I think we're all quite aware of the placebo effect. Lots of interesting exploration into this over time. In particular, as we consider NASH specifically, two areas that may have substantial interaction with clinical trial outcomes are, of course, dosing compliance, as well as weight gain or weight loss. Whenever there are those sort of unexpectedly substantial placebo responses, it makes it very difficult to establish that there was true pharmacologic efficacy in the treatment cohorts. And in fact, unfortunately, there's a very recent example of this. Elafibranor in its phase 3 trials, GenFit issued this press release about a year ago now, basically reported a situation just like that with 72-week results. They basically showed no difference in the primary or secondary endpoint of comparing treatment to placebo. And by and large, it seems that I think common sense prevails. The placebo response was substantial in terms of fibrosis improvement of 22% of the patients in the placebo group that wasn't on treatment. So one question that always comes up was, is there a chance that group of people, the placebo cohort, did something different? And so we can apply Qsp modeling to explore some of the differences. I don't think dosing compliance will help much with the placebo group, but was there the potential for something like weight loss in a specific cohort that may have influenced these results? And so, in fact, we are able to do that. But before we dive into that, let's just point out that body weight is indeed associated with both kind of the progression of NASH as well as the reversal of NASH. And by that, I mean with weight gain. If you look at the data from Wong et al. On the bottom, you'll see that there's often a correlation that's established between weight gain and a worsening of the NAS score as well as fibrosis. So there's really seems to, and mechanistically, we know that the weight gain imposes a greater lipid burden upon the liver, and everything else kind of follows from there. Working in the opposite direction, weight loss relieves that lipid burden as the adipose mass declines, as food consumption decreases and substrate availability to the liver declines. The lipid state and everything else, the improvement is certainly in place. Again, we have all these mechanisms established within Naflsim, the ability to simulate then independently the effects of weight loss or weight gain, as well as the effects of treatments such as those just described by both Zach and Christina. And so again, just to kind of show that in fact we are on target when it comes to the weight loss and the effects. Two nice data sets published in relatively recent years, Bilar Gomez and Hamid, have shown that with weight loss over the course of 1 to 2 years, relatively modest amounts, roughly 5 to 10%, is indeed associated with improvements in the NAS score. And it seems to some extent improvements of steatosis, inflammation, and ballooning. And if you look at the clinical results in the black, and the simulation results of our sim cohorts, there is a really nice agreement between those, all of the above, the individual components as well as the aggregate NAS score, and kind of really, again, providing confidence that Naflsim is indeed able to predict the response due to weight loss. Taking that a step further, again, to one of the other primary outcome, often the fibrosis. And so this is also reported in the Villar-Gomez paper. This is a little more complicated. Let's see if I can walk you through it. Basically, what they saw is throughout their clinical cohort of the patients that had fibrosis, and again, that 1 to 2 year period of 5 to 10% weight loss, they saw that about 38% of the patients had a reduction of at least one stage of the fibrosis score in response to the weight loss. 44% of the clinical patients remained stable, whilst 18% in fact increased a fibrosis score. Our sim cohorts really showed remarkable similarity, particularly if we look at the stabilized group, almost exactly the same proportion, 46% in the sim cohorts. And I would argue we're very much in the same ballpark with respect to the regression, where 54% of the simulated patients showed a reduction of fibrosis score of at least 1 over that same period of 1 year. So this is, with these simulation results, I think it's very clear that we're able to establish the ability to have weight loss impact some of the primary outcomes that are relevant. And so again, we can run simulations with weight loss present, in the absence of weight loss, with drug on board, in the absence of drug on board, to varying degrees of all of the above, and get a sense for if there's an interaction effect between the treatment and the weight loss, or weight gain and treatment, or if there's a potential for a kind of a divergent response in the placebo group relative to the treatment cohort. And that may help kind of unobfuscate some of the results such as those reported by GenFit. So let me just conclude before we move into the Q&A section by just pointing out that, as I mentioned before, all of our offerings, in this case, specifically NAFLDsym V2A, is indeed available for licensing. Another very important point is that with all of our QSP models, the equations can be viewed by users and potentially be modified by users. That affords us the flexibility of introducing additional mechanisms and the ability to simulate additional compounds. And speaking of us doing this, in fact, we are able to do services type projects in order to evaluate all of these. We do it in a sort of a staged way that allows everybody to be on board and apprised of progress. Of course, obviously, ultimately, the cost of each project are dependent on the resources, the scope of the project, et cetera. Finally, certainly you've heard from Christina and Zach today, but truly we stand on the shoulders of those who came before us. A fantastic team of individuals have contributed to the development of NAFLDsym and our other offerings. It's an interdisciplinary, ranging from the mathematicians to the engineers, to the pharmacologists, to the immunologists, to even me, the nutritional biochemist originally. So with that, I'll conclude by thanking you for your attention, and we can move into the question and answer section of the session.
Great, thanks, Scott. And I will invite all of our panelists to come on camera so we can answer the questions. And the first one in the queue, Scott, is for you. How does Naturalism estimate NAS and fibrosis stage score with simulated biomarker change mathematically?
It's a great question. So let's start with the fibrosis score. It's actually, I think, a really interesting approach that we've taken. When you look at how the simulation approach abides by the measurement approach, which is an individual reads the slides and they say, oh, there's an excess of fibrosis in this particular region of the liver, hence assigning a score of 1, 2, or 3. But the critical component is that there is an excess. It is something of a subjective measurement, i.e. an expert eye is able to discern that, someone who had, underwent a great deal of training. So in order for us, we can't simulate that expert eye necessarily, but we can simulate the difference between what, normal and too much, if you will, that excess. So in fact, that's what we do. We, in parallel, simulate the amount of collagen that would be there in the absence of disease, the amount of collagen that is there in the presence of disease, in comparison of the two, which allows us to then basically provide that kind of discernment that the expert eye would do as well, looking at a histological slide. For the NAS score, it's not quite the same. Basically, we are computing, as you saw, inflammation, ballooning, which is a measure of apoptosis, hepatocellular apoptosis, and steatosis, which is the liver lipid and the accumulation thereof. And so we basically translate our specific results of and specific calculation of those into the histologic measurements, and then aggregating them all together to provide that NAS score.
Great, thank you. Christina, this one's for you. Why did CVC treatment did not work? What needs improvement? And is it the potency of the compound or the pathway itself? Oh, you're on mute.
Thank you for that. I'm actually glad someone asked this question, because this is something that we've been wondering ourselves. So we're actually running simulations, changing the potency, seeing if changing the potency will affect the pathways a little stronger and have better outcomes for our patients. And we're hoping to present those results at a future conference. So unfortunately, I can't say right now, but at conferences, that DILIsym typically attends, be on the lookout. We will present those results as soon as possible.
Great. Ravindra, check out our events page, and we'll post on there what conference we'll be at with those answers. Thank you for joining us today. Scott, Zach, this one's for you. You modified the parameters or models in Naflsim to fit the clinical studies presented in your presentation. Or did you not modify the parameters or models?
Yeah, it's a great question. And the brief answer is the diagram that Scott went through at the beginning, we've calibrated some of the sub-models for liver fat, inflammation, and fibrosis. So those models are are there and calibrated to data. And then what we do for a treatment like CVC or the one I discussed is we'll look at pathways that are existing in the models that we want to inhibit, and we'll modify equations to inhibit or enhance certain pathways. And so there will be equation modifications there, and then additional parameters that we might calibrate. So really, it does involve some additional equations and parameter optimization.
Okay. Christina, back to you. Our next audience member wonders if there's any change in serum adiponectin level in the CVC simulation in your first case study.
Sure. So we did look into this. It was one of the outputs that we had plotted initially. We just didn't have time to share it today. We didn't have any significant changes in adiponectin. I do know that they reported adiponectin as an output in the supplement of their RATZU 2020 paper. And they saw minor differences, so a minor decrease in the adiponectin, but it was the same kind of story where you saw it in tenecrevirac treated patients and you saw that decrease in the placebo group as well. So it seems to be kind of a wash across the board, and we predicted minimal changes as well.
Okay. Scott, is it possible to predict selected population effect from the clinical study, which we have the data for?
It is indeed, but let me be clear that this is to delineate between, say, a classic PK/PD type analysis, which this question sort of, I think, is describing, versus a QSP. Sometimes I use the analogy of a puzzle across where you'll take the pieces and it comes together to form a full image. In the PK/PD, oftentimes you're using the mathematics, the modeling, to kind of pull apart those pieces and understand what had happened, how they might have come together. With QSP modeling, you actually go in the reverse direction. As Zach described, we have those mechanistic components, and we put each of those pieces together to form that overall integrated picture, and we understand mechanistically how the components come together. So in order to understand, then we do some modifications and generate our SymPops to introduce the variability. So we are able to then, with that mechanistic modeling throughout our entire 1,700 simulated patients, understand if there are subgroups that have specific proclivities, specific characteristics that might encourage a response to treatment or a lack of response. So again, I just wanted to kind of just describe the difference. Both approaches, methodologies allow for such an analysis, but coming at it from, I think, different directions.
Good. I like the puzzle analogy. We have time for a couple more questions. So Zach, this one's for you. In addition to the examples provided during your presentation, have you supported the development of compounds in first-in-human clinical studies or phase 2 or phase 3 studies?
Yes, yeah, we've supported across the development spectrum, and then also, in addition, we've looked at target engagement, so which is a little closer to the first-in-human. So looking at, say, again, inhibiting a pathway and looking at 25%, 50%, 75%, and so that can be a little more exploratory, but also very informative. But yeah, across those different stages, we've supported compound development.
Great. All right, Christina, last one for you, and then Scott, I'll let you have the final question. So Christina, how many different targets can you simulate with Naflsim?
So I think there's really not a limit. As Zach just said, right, we can kind of explore any pathway. You can put an effect on any pathway that exists within NAFLDsym, or we could add additional pathways that aren't there given the right amount of data. So there's not really a limit for the number of targets we can simulate. I think we've probably simulated anywhere from 12 to 18 different targets in probably 25 or so compounds now. So there's not a limit, and we are eager to do more, for sure.
Good. Okay, so Scott, last question. Is Naflsim able to predict reductions in fibrosis score in as short a period as 12 to 16 weeks as it's recently been reported in literature?
Well, I'm so glad you asked that question, Arlene. To me, this is, I think, one of the most exciting developments in the NASH area. I'll recall when I was a graduate student studying kind of the liver and fibrosis, and it was basically said that the dogma was, if you have fibrosis, it is irreversible. And then I think our understanding evolved a bit as hepatitis C treatments became available. And heck, it is reversible, but it takes a while, or maybe a couple of years. And now in late 2020, early 2021, we have a couple of reports of NASH compounds in phase 2, phase 3 studies, 12 to 16 weeks to have as many as 40 to 60% of the participants have a reduction in fibrosis stage in 12 to 16 weeks. Super exciting. And also made us ask that very same question. So we ran the simulations, and sure enough, depending on the treatment and the effectiveness of it, in fact, we are able to as well predict reductions in fibrosis scores in that shorter period of time. Really, again, remarkable, an evolution of knowledge. And I think that's also one of the great things of Qsp models is it's a synthesis and aggregation of all that knowledge. And the predictions, yes, in this case, certainly lined up.
Amazing, exactly, an evolution of knowledge. Well, with that, thank you to Christina, Zach, Scott, thank you. We appreciate you sharing with us how Naflsim can help clinical NASH treatment programs in a number of ways. And thank you for everyone who submitted your question. If yours was not addressed, we will follow up with you in an email. I do ask that you take the 1 second to answer the poll question at the end of the survey when the webinar closes. And if you missed any part of the webinar today, it has been recorded and will be available at www.simulations-plus.com for an on-demand replay. This concludes our webinar for today. Have a great day. Thank you, everyone.